Okay, hi everyone. This is our final exam live stream review. Um, Feel free to ask whatever questions you have, and I will answer them, and we'll just go from there. Um, let's see. While I'm waiting for people to ask questions, I'll just remind you of the format of your exam. The format of the exam will be all multiple choice, 100 multiple choice questions. So, kind of similar to your exams, just a lot longer. And then you should expect to see a lot of different topics. Remember that for my students, about 45% of your exam is genetics. So you definitely want to make sure you understand all the different types of crosses you're making, how to do a Punnett square. Make sure you remember when you are doing the Punnett squares to ask yourself, is this an autosomal trait or is this a sex-linked trait? Because if it's an autosomal trait, you would do a normal Punnett square. But if it is a sex-linked trait, hi, Janya, um, then you would need to put the X and the Y into the Punnett squares, right? Okay, so go ahead and ask any questions. I guess if there aren't questions, I'll just start talking about random stuff. And hopefully our interruptions will be to a minimum here from the kiddos. What are some things that you guys want to know about? What are some things you're not sure about or you have questions about? Anything? Let me see. What can I talk about while there are questions? Or while I wait for questions. I wanted to watch this. Let's see. Do make sure that you're thinking about, oh, okay, a rule. What's the difference between homologous chromosomes and sister chromatids? Okay, so think of it, remember, as I'm going to use a human cell as an example, but it doesn't have to be human, right? Most of your cells in your body are diploid, and that means they have 46 chromosomes or 23 pairs, meaning of every chromosome, I have two of them. One came from mom, one from dad. So if we give them like a number, like one, number two, number three, all the way to 23, right? That means they have two number ones, two number twos, two number threes, etc. And those two number ones and those two number twos are homologous chromosomes. Okay. But when you are preparing to divide a cell, you need to make sure you're going to have enough DNA. And so, in the process of getting ready for a cell to divide by mitosis, you need to duplicate the DNA inside that cell. And that's when your chromosome goes from one chromosome that it consists of one copy to the chromosome is now made up of two sister chromatids. Um, so hopefully that kind of helps to sort of think of it differently, right? Is that your homologous chromosomes exist in your cell at all times. Whether or not they're actually pairing up with their counterpart is the question. So in mitosis, you have homologous chromosomes. It just doesn't matter that you have them. Like they never match up with their other chromosome. Like the two number ones never get together and pair up, right? That only happens when you're preparing to go through meiosis. So hopefully that helps. Think of also the homologous chromosomes as one from mom, one from dad. Whereas sister chromatids are like identical copies of the same chromosome. Okay, what other questions have you got? Anything? Remember also, for my students, at least for meiosis, I don't need you to know all the individual details. I want you to know the big idea, which is homologous chromosomes pair up, they cross over, the pairs separate at the end of meiosis 1. Now you have two haploid cells, but each haploid cell has one chromosome that's made of two sister chromatids. So then you divide again to get individual cells, four haploids, which have only one copy of a chromosome in them. When I say light reactions during cellular respiration, there are no light reactions in cellular respiration. Cellular respiration is glycolysis, the Krebs cycle, the ETC. The light reactions is only for photosynthesis. Now, there is a link between them, right? Because if a plant does the light reactions, traps the sunlight's energy, it's going to use the energy that it traps from the sunlight to make ATP and NADPH. that ATP and NADPH is used to make glucose. That glucose is then what goes through cellular respiration, but there's no light reactions in cellular respiration. Let's see. Make sure that you can tell the difference between certain terms, right, that are going to kind of run together. So when we're talking about genetics, make sure you can tell the difference between things like a gene, an allele, and a chromosome. They're like different levels on some level, right? Okay. Grace is asking, what are keystone species? Are they the top predators or producers in an ecosystem? They don't have to be. Keystone species, by definition, are organisms that in their ecosystem they play such an important role that if they were gone, then they would have a major impact on the ecosystem, right? So like they could be the top predators, they don't have to be, but it means that their role, whatever it is so pivotal, and usually they play more than one role, that if they were to be gone, then now they would basically potentially cause the collapse of the ecosystem. So wolves, which are a top predator, could be, they are considered a keystone species in that particular ecosystem, like in Yellowstone. But for example, prairie dogs, which are like little rodent-like things, they are not the top predator in the ecosystem, but they perform so many different roles in the ecosystem that if you take them out, they really mess up the ecosystem. Let's see. When I ask about cell organelles, do I mean the ones in plants or animals? I mean both, right? I mean, there are a lot of organelles that plants and animals share. And then there's obviously the few that they don't share, namely the chloroplast, which is what the plants have but animals don't have. What is it called when the chromosomes mix up the DNA? I think what you might be talking about is crossing over, Anita. That is during the beginning of meiosis, and that's just to sort of give you more variety in your DNA, that you don't have all the same combinations in the sister chromatids. But essentially, what happens is the sister the homologous chromosomes get together next to each other. They form something called a tetrad, and then part of them breaks off and switches with the other piece. And in doing so, now the combinations of alleles you have are different for the for each sister chromatid. So actually, at the end of meiosis, you no longer have four identical. The haploids are not the same, right? And that's why each haploid can become a different gamete, which then could become part of a different offspring. Okay. Other questions? Yes, the only haploid cells in our body are sex cells. Correct. And those originate from diploid cells. So there are certain diploid cells in the reproductive organs which will go through meiosis to form the sex cells, the gametes, the sperm and the egg. Let's see. What else? Yes, Grace, you are correct. That is crossing over. Let's see. What else can I remind you of? Make sure you know things like osmosis and diffusion, right? But remember, don't just memorize the definitions. Make sure you can do problems with them. How does the nonpolar tail and polar head work? Arul, I assume you mean the phospholipids. So remember, in a cell membrane, you have phospholipids, and the phospholipid is really unique because it has a the polar head, which is phosphate, and then the nonpolar tail, which is which is the fatty acids. And so the idea is that that creates a physical barrier that to make up the cell membrane. So only things that are small and nonpolar can really pass through that large nonpolar middle portion. And that's what gives the cell membrane its selective permeability. Yes, endocytosis and exocytosis are both active transport. Hi, baby. So they do require energy. Let's see. Make sure you know your biomolecules, right? Make sure you know how to recognize them in terms of like both visually, but what they're for. Those are going to be important. Let's see. What else? Are you guys comfortable with pedigrees? Do you guys have any questions about those? Or enzymes, right? You want to make sure you remember different aspects about enzymes, things like vocabulary. So, like the enzyme is a protein, the thing that it works on, or things, I should say, because it could be more than one thing, would be our substrates, it's their reactants. The enzymes, what they do is they catalyze reactions. Okay, is the ETC thing yeah, essentially, I mean, so what the ETC actually is it's a bunch of proteins in the cell membrane where as the electrons get passed down that membrane, it uses the energy in the electrons to make ATP. In the story, we told it as it squeezes all the energy out of the electrons, but that essentially what it is the electrons as they are passed from protein to protein um, cause the cell to make ATP, a lot of it. So it's kind of like cashing in your NADPH, no, it's not your NADPH, it's your NADH and your FADH2s, right? Because you generate all this NADH and FADH2 mostly in the Krebs cycle. And now as a result, you need to, but that's not ATP. So it's sort of like cashing out at a casino where now they're going to trade in all that NADH and FADH2 in the form of ATP. But yes, and it's actually a similar thing in the photosynthesis. I mean, in terms of what it is molecularly, both create ATP. Okay, let's see. Make sure you understand the structure of leaf. Make sure you know the difference between plant cells and animal cells. What else? Other questions you guys have? How many parts do we need it's not a matter of like number of parts. I would say I want you to be able to look at the basic picture of a leaf and know some of the major parts that are involved with cellular respiration, photosynthesis and cellular respiration, right? So namely with the leaf on the outside, you have the guard cells, which are the controlling things that let the stomata be open or closed, right? Um, that's going to be the that what allows the gases to come in and out, and that's going to play into photosynthesis. You would want to know where the photosynthetic reactions would happen, which would be in the palisade and the spongy mesophyll cells. Now you need water for all of that to happen, so you have to have a vein, which part of which is the xylem, and then the xylem is going to bring the water, which you need for photosynthesis. If you study everything on the yes, but remember that the salmon sheet is topics. Like it's not going into necessarily specific details, right? But yes, if you study all those topics, you should be fine. But then you have to kind of make sure you're covering each topic fully. My students know I'm not collecting your notebook on the day of the final. You have enough to worry about, so we're just going to do finals. Bring your study guide to turn in for credit, but no journals. Let's see. So other things you guys want to know about? Let's see. Blood typing. I noticed that some people had a lot of, not a lot, but there were some people who had trouble with that on your quiz. So, okay, so let's see. We've got a question. What's the inheritance of sex-linked traits? I'm not sure what you mean by that. Essentially a sex link, but I'll try to answer it and hopefully get to what you're asking. So a sex-linked trait, remember, means on the X or Y chromosome. Now, more often than not, we're talking about a X-linked chromosome, just because the ones that are on the X. Oh yes, and I will get to water in a second. So okay, so let's finish with sex-linked traits. So with a sex-linked trait, what it means is remember we have, so in terms of vocabulary, we have 46 chromosomes, right? 22 pairs of those 46 are what we call autosomal chromosomes, meaning it doesn't matter male or female, they're all the same. My last set of homologous chromosomes are my sex chromosomes. And even though they do define an individual's um, sex in terms of male or female, they also have other genes on them, like color blindness, clotting factors that have to do with hemophilia. So, in that sense, if a gene that you wouldn't necessarily associate with males or females is on the X or Y chromosome, then it's sex linked. What that means then is since females have two X's and males have one X, it kind of changes the likelihood, especially if it's a recessive or disorder or trait, of males versus females getting it, right? Because males only have one X chromosome. Pretty much whatever X chromosome they have is the trait they show. There really is no almost, there's no masking it for them, right? Whereas for a female could have a be heterozygous for a trait and not show the trait, but be a carrier. But males can basically never be carriers for an X-linked trait or sex-linked trait on the X chromosome because then it wouldn't be able to. They only have one X. So we see it usually a recessive disorder that's on an X-linked the sex-linked trait tends to be more common, like color blindness, in males and females. Doesn't mean females can't have the trait; just means they don't have as high of a likelihood of having it. Okay, so let me go through the questions I'm getting here. Can I explain how water gets to a house? Yes. So assuming that your water, well, we can track it from any given point, right? But let's just say we track it from the sky. There is water vapor, right? It condenses, it precipitates, it falls down into a number of areas. One place it could fall could be a reservoir. It could fall down into the ground and soak into the groundwater, which would be in an aquifer. In which case, now either from the reservoir or the aquifer, it gets sent or pumped to the water treatment plant. In the water treatment plant, it gets cleaned out, and from there, it would go through pipes to your house. Now, once you're done using that water, it goes back down the sink. It would be sent to the wastewater treatment plant to get cleaned out before it's ultimately dumped in the bay. And it sort of now completes back that cycle, right? Because now water that's in the bay can evaporate, go back into the atmosphere, and that too can now condense and precipitate, and the whole thing can start over. Let's see. Okay, someone asked, when do we use meiosis instead of mitosis? So remember, think about the purpose, right? Meiosis is to create gametes. So meiosis would really only happen when the purpose or the goal is to create haploid cells. Whereas mitosis is going to have a much broader usage because when you're growing, you need more cells, you do mitosis. When you get like a paper cut and you've damaged cells and you need to replace them, you do mitosis, right? So there's a lot more times, I guess you could say, when you're going to potentially need to make more cells that are just like the ones you already have than when you would be making a bunch of gametes. Let's see. Where does water go from the Sierra Nevada snowpack? So the Sierra Nevada snowpack is essentially like stored water, right? The idea is that it gets frozen in the wintertime and stays frozen. And then now, as the weather gets warmer and that ice and snow starts to melt, it starts to flow. And when water flows, it's going to flow into like local rivers and streams. It might also soak into the ground or replenish the aquifer. So the idea is then it's going to add to the water source that we have for drinkable water that could be sent to our water treatment plant. Okay, other questions? Yep. Okay, so yeah, it looks like you guys have all the right answers as you're answering for each other, so that's good. Let's see, what else? I should probably, for my students, just define it really quickly. We never called it this, but I put it in my my paper there. A monohybrid cross just means like a one trait cross, which is your standard Punnett square. What are the stages of meiosis? So, Arul, remember, I don't actually want you to know the stages of meiosis. I intentionally had you kind of like not focus on the names of them. So, if we're talking stages, I would think like major events, which would be before meiosis happens, the chromosomes duplicate. So now every chromosome is made up of two sister chromatids. Then the homologous pairs would get together and crossing over would happen. So you kind of mix up the combination of genes. Then in meiosis 1, homologous chromosomes separate. And then in meiosis two, sister chromatids separate, so the end result is the 4 haploid cells. Yes, the Krebs cycle and the citric acid cycle are the same thing, so um, you can consider them one and the same. Um, Aqueducts. So you may have actually seen aqueducts before, like when you're driving. If you've been ever driving, like from the Bay Area down to LA, then essentially aqueducts are these really large, usually open air channels where the water can flow to get from like one location to another. So you might, for example, take like water from a reservoir and you would take it through the aqueducts to like a water treatment plant. So it's a way to transport the water sort of on its way to the water treatment plant. Um, they do tend to be open air, like mean they're open to the outside. So that's why, like when it rains, for example, then water can fall into the aqueduct as well. Um, and yes, that is so. Thank you, Grace. It looks like we answered the question on water exiting the house too. Okay. All right. So what else? Let's see. Yeah, you can work on it. You can totally act. You mean your study guide? You can put whatever you want on it. I mean, that's fine. It's like it's just that once you go into the final exam, you'll turn it in to me at the end of your final exam. But you're certainly welcome to do whatever you want to do with it. Okay, the natural versus constructed waterways. Um, the natural water cycle is just the idea that, like, if humans weren't involved, what would happen, right? So that would be like okay. Um, so the natural water cycle would be like evaporation, condensation, precipitation, right? Just the stuff where the water kind of goes around and around. Oh, plasma membrane. Yes, I'll get to that. Sorry. Um, Whereas the constructed water cycle means when humans get involved, right? So now it's when we add in things like, taking water to the water treatment plant and the sewage plants and all that stuff. Um, Yes, again, there's the salmon exam topics would be fine, but topics is still kind of general, so you want to make sure you're covering everything within those topics. Okay, really quickly, let me answer the question about mitochondria. I think ribosomes, yes, are smaller than mitochondria and chloroplasts. So the plasma membrane, again, remember, is the idea of your cell membrane. So plasma membrane, cell membranes, the same thing. And so that one, the main thing to know about the cell membrane is that it is selectively permeable, meaning um, not everything is going to be able to get across the cell membrane, right? The cell membrane is made up of a phospholipid bilayer, so it's 2 layers of phospholipids. They're pointing such that we always show them like kind of like the fatty acid tails, kind of like that, right? So the tails are pointing inside, and the phosphates are on the outside. So the tails themselves are nonpolar. So what they're really creating is like this nonpolar region in the middle where, unless you are also nonpolar, because remember things only go through if they're like that area, um, then you effectively can't have things cross the membrane. Now, also embedded in the membrane, though, are things like protein channels. There are carbohydrates on the outside, and there's cholesterol, which contributes to the fluidity. So, in terms of the cell membrane, that all of those things in combination are what make the plasma membrane fluid, meaning things are still moving around, but have a certain permeability that not everything can just cross. Okay, let's see. Aquaporins are basically specific protein channels for water. So water can cross the cell membrane in 2 ways. It can just cross, um, and then, or it could also like go. So it can go through like simple diffusion, or it could also go through an aquaporin. Would a plant die if you took out its chloroplast? Um, yeah, essentially it would, right? Because plants have no way of eating. So, unless you had a way of somehow feeding the plant or giving it glucose, then it would die because it would never have any glucose to use for cellular respiration, right? We get glucose by eating it. but a plant doesn't do that. And so, unless they have a way to get glucose, and normally they make it themselves through photosynthesis, then yes, a plant would die. So, yes, carnivorous plants actually do photosynthesis because carnivorous plants, when they eat insects, they're actually just trying to supplement their diet. They're getting protein from those insects, but they're still doing photosynthesis. It doesn't replace the glucose that they're getting. Um, let's see. So factors that affect the health of a creek are going to be things like, and I think we took notes on it, so look in your notebooks. Things like the turbidity, so how cloudy the water is, things like pollution, so like acid rain. All of those things are going to contribute to the health of a creek. Also, like the trees on the side bank, right? Because if you have like not as many trees, then the soil in the on the side of the banks kind of goes down into the water a lot more, and then that's also going to affect like how cloudy the water is, which would affect like how well the plants in there can get sunlight for photosynthesis and things like that. um let's see, I don't know if I missed anything, so tell me if I have. um but it looks like you've got that. Okay. Let's see. Oh yeah, so how does a flower emit smells? Um, Essentially, a flower emits smells because it is producing some sort of molecule that has a smell to it. It's possible that that's a protein, right? So it just could be something, somehow a plant that, and it's, it could be evolutionarily a way to attract bees and things over that would help it pollinate, which would help it reproduce. Um, what's everything? Okay. Um, in terms of everything, what I want you to be able to know about waterways is I want you to be able to trace the pathway of water, both in the natural and constructed water cycles. I want you to be able to tell me where water is going or coming from. I want you to be able to tell me about non-point versus point source pollution. Um, I want you to be able to essentially explain sort of where how water flows and what the issues are when, we overdo it, right? In terms of like taking water out, etc. Um Organic, so resources of carbon just mean things that are made of carbon. Organic matter means anything that contains carbon. So we are made of organic matter, right? Trees are made of organic matter. Again, essentially anything again that has carbon in it, we consider organic. It's a different kind of organic, not like the organic you hear about, like in terms of the supermarket. That has more to do with like no pesticides and things like that. When we talk about it in a biological sense, organic means to contain carbon. Okay, the carbon cycle, and so carbon is what makes something organic, right? So it's not that carbon is organic. It is organic matter, but it's the major defining element of something organic, right? So like um wood that makes up your table, that's made up of organic matter. The orange you eat for lunch is organic matter. The carbon cycle is looking at how carbon then moves, just like we had the water cycle. Remember, we can also have carbon. So things like combustion release carbon into the atmosphere, right? Trees and photosynthesis are going to absorb carbon. So that's a carbon cycle, like looking at how carbon moves around. Um, the slow carbon cycle and the fast carbon cycle. So for my students, we didn't really go over that, so I wouldn't worry too much about it. It's just the idea that um, basically some carbon cycles through really, really slowly. Like it gets trapped and it can be down in in fossil fuels and stuff for like thousands and thousands of years. And then other carbon is like really quickly overturned, right? So that's sort of the idea of slow versus fast cycles. But for my students, I didn't go over that, so I wouldn't worry about that. um, Sustainable means that it's something that we can sort of have to keep going, right? That if it's not sustainable means you could use it up and it's gone, like there's nothing more. So we want to kind of make sure that the resources we use are sustainable, so that we are not just using something up and then it's gone forever. Okay, let's see. Oh, is polarity basically how attractive? No, polarity really has to do with what's going on inside the molecule. So remember, let me see if I can find two
Mommy,
demo
let's
molecules.
stop the show.
Okay, you guys, you're okay, right? Okay, so let's say I had like two atoms. We'll just call them this, right? Let's say this is oxygen and this is hydrogen. The idea is that depending on the atom, how much they pull on the electrons differs. So if they're sharing equally, so the bond, first of all, within a molecule is a covalent bond. But how they're sharing those electrons in a covalent bond depends. So if it's a polar or polarity has to do with the idea that like one atom pulls on those electrons more strongly than the other one, which means if the electrons, let's just pretend these are the electrons, right? The electrons then, instead of being sort of directly in the middle, are going to be closer to one atom than the other atom. And because of that, because electrons have a negative charge, that makes this molecule slightly negative, this one versus slightly positive, just because it's a lack of electrons versus electrons, right? That's sort of the general nose I want to. Yes, you may. Um, so, the bonds I need you to know are just basically covalent, which is bonds within a molecule where they're sharing electrons, whether that's equally or unequally. Ionic, which is where they're not sharing and one atom pretty much grabs them from the other. And then the important thing about polarity, especially as it applies to water.
Paw Patrol.
Okay, you have to watch it, then I can't change it right now, okay?
Can you please? Can you just please say 1 second?
I know, you can bring me the remote and I can try and change it from here. Okay. Um, so the idea with polarity and the reason it's important is because when you have a polar bond, right, and you now have this slightly negative, we'll make these electrons like slightly negative, right? That means this slightly negative part could be attracted to a slightly positive part of another molecule. And that means that
Mommy,
you'll be able to form hydrogen
use
bonds.
the remote.
Thank you, Sammy. 1 second.
Me too, I can watch another butter beans.
Okay.
No, butter beans.
Sorry, give me a second here. Okay, Sammy, I just have to pick this one for now, okay? And then when Daddy comes home, we'll do it. Okay. Sorry, guys. So a hydrogen bond then forms between waters because each water, think of it, has like a each water basically has like a slightly negative part and a slightly positive part, so they can be attracted to each other because of that, and because of that you'll have hydrogen bonds between water, which then causes the property of cohesion. Reclaimed means it's water that's already been used, but we're like going to reuse it. So usually we don't want to use it for things like drinking because it's not fully cleaned out, but it's still something we could use like for watering our plants, for example, things like that. When we talk about electrons, why are they just like around? Shouldn't they be? I mean, they are in an electron cloud, but we don't really talk about it too much in that context. Like I just need you to know that what they are very generally, and you'll get a lot more of that in chemistry if you take it next year. Gray water and reclaimed water, I think they're similar. I'm sure there is a slight difference in the definition, but I don't need you to know them, any sort of specifics there. But the idea is that they're usually, like not fully, totally clean water. Like you wouldn't drink either of them. Okay. Let's see. I don't know if we got everything here. Let's see. So yes, so when they say humans are carbon-based, it means that we, at the core, what we're made up of is a lot of carbon, right? I mean, almost every molecule in your body has some carbon in it. So gray water, I mean, yeah, I mean, I barely talked about gray water. I wouldn't worry about that. So I would say let's not, spend too long on it. Let's see. know that it can't be recycled. I think it's just that it's not, like I said, it's not drinkable. So we can use it for other things, but we're not going to use it for, we're not going to put it back in someone's house, right? Because tap water is actually pretty tightly regulated, comparatively speaking. Like it's actually, I think, more regulated than like, bottled water and things like that. Let's see. Looks like someone's asking something, but I can't see it. Let me see. Can atoms get bigger or smaller? No, atoms don't get bigger or smaller. I mean, atoms are atoms. Essentially, without getting too much into the chemistry, atoms are defined by, the a number of electrons that they have. And so, no. And so an atom really is like, carbon atom, oxygen, hydrogen, those are different atoms. And so they don't get bigger or smaller. They are a certain manner. CHIPO represents the threats to biodiversity. So C stands for climate change. H stands for habitat fragmentation, right? I stands for invasive species. P stands for population. The other P stands for pollution. And then the last one, the O stands for like overconsumption or over exploitation. The difference between NADPH and FADH. Well, first of all, in terms of what the ones you mentioned, they are the difference between them is they're all electron carriers, so they will receive electrons during the processes of photosynthesis and cell respiration. NADPH is actually only in photosynthesis, and FADH is in cell respiration. So I guess you could say differences between them are they're involved in different reactions. They're physically different molecules, but they do the same thing, just in different reactions. So FADH is receiving electrons during. They aren't protein. I think they're more complicated. I mean, they have these really complicated names like nicotinamide something. So I don't think they're proteins. No, I mean they're molecules. I'm not sure what exact classification we'd put them under. So they could be a protein, I suppose. But I actually don't remember the exact molecular structure. So just know them as electron carriers and it's sufficient. Let's see. Remember, there are sort of like different forms, I guess you could say, of the electron carriers, right? The empty carrier that doesn't have the electron is like FAD, NADP plus, NAD plus, whereas like the full one after it's received an electron be NADPH, FADH2, and NADH. A vesicle is basically like a carrier, like a bubble carrier, right? Imagine that in order for cells to transport things, they basically put them in these vesicles and they move them around the cell, but also to get it out of the cell through a vesicle. So it's kind of like a bubble carrier within the cell. And it's oftentimes how you're going to transport things from one part of the cell membrane to another. Or not cell membrane, one part of the cell to another. But it could also be to get something out of the cell. Let's see. Over harvesting, over exploitation is actually kind of the same thing. I mean, it just depends what you want to call it. It's all the same idea, right? If we over exploit something, we are probably also over harvesting it. So it's just the idea that we're sort of taking out too many resources and potentially then damaging the ecosystems that they're a part of. So, I mean, I wouldn't stress about it. Some teachers may have taught it as over exploitation. Some teachers may have taught it over harvesting. It's the same thing. Two names for the same thing. Let's see. Okay? So the purpose of really any fermentation is to keep glycolysis going, right? Remember that comparatively, cellular respiration makes you a ton of ATP. But if you don't have oxygen, you shut down the second and third parts of the cellular respiration process, right? Glycolysis, the Krebs cycle and the ETC, they can't continue. And if you can't continue, then you're barely going to make any ATP because you're normally going to be cashing out all those NADHs and FADH2s in the ETC. So what alcoholic fermentation does is it basically lets you tread water. It lets you like keep going with just glycolysis. But if you're going to just keep glycolysis going, you have to keep giving glycolysis the reactants it needs. And one of the reactants it's going to need is NADH, or an NAD plus, I should say. Right? Normally you'd get that NAD plus as you empty out the electron carriers in the ETC. Since you're not doing the ETC, you have to find a different way to get those electron carriers emptied. So in doing so, you are going to, so alcoholic fermentation takes that pyruvic acid, turns it into CO2 and ethanol, and in doing that regenerates your NAD plus to allow it to do more glycolysis. It's the same with lactic acid fermentation. It's just you go different reaction, but the goal is the same, which is to remake your NAD plus to keep glycolysis going to make a little bit of ATP. No, the smooth and rough ER are quite different because they do really different things, right? I mean, they probably look fairly structurally similar. And yes, the rough ER has ribosomes on it. That's because the rough ER is the one involved in proteins and protein export. So rough ER is going to be the one that modifies the proteins and sort of those steps to get it ready to exit the cell. Whereas the smooth ER is not involved in protein transport at all. So the smooth ER is going to do things like synthesize lipids, which might be, for example, the cell membrane, but also things like the detoxification of things of the cell, right? Because if there are toxins that come in, the smooth ER is going to take care of those. So the really the only thing they have in common is their name. They don't do similar things at all. And I would say that was one of the really common mistakes I saw on the second exam, is people kind of mixing up the two of them. So yes, the rough ER is rough because it has ribosomes. So you want to know that also it's involved in the process of protein exporting or protein transport, right? Because it's modifying things. Okay, what else? Anything else? Let's see. Have we caught everything else? Oh, what kind of diffusion does not need energy? So really, all diffusion doesn't require energy. Whether it's simple diffusion or it's facilitated diffusion, that all falls under the umbrella term of passive transport, which means it does not require energy. It's always things moving from an area of high concentration to an area of low concentration, whether that's directly through the cell membrane or if it's going through a protein channel. It's always going to require no energy. Does lactic acid fermentation take place in all organisms other than I would say no, not all organisms, right? I mean, humans do it. That's sort of the main example I give you, and probably other organisms as well. But the main example that we give for it is humans. What is the path of ribosomes? Ribosomes don't have a path. I mean, ribosomes themselves, their job is to make proteins, so they don't have a path because they're just there. Now, where could they be? It depends. So I suppose if you're talking about it, you could say that a ribosome's path could be that it's made in the nucleolus and then it will exit there and go to the cytoplasm, where it will either be free floating and make proteins, or it could be a ribosome that's attached to the rough ER, in which case the protein it makes will now be one that's going to leave the cell. Once that protein is made, it goes from the ribosome into the rough ER, from the rough ER to the Golgi, and the Golgi it will now exit the cell. Is it possible for something to be biotic and abiotic? No, because by definition, something that is biotic is something that either came from a living thing or is itself a living thing, right? But something abiotic, is from a non-living thing. So no, I would say that no, it cannot be. Is anything that is or was living biotic? Yes. So like dog poo, because it came from a living thing, is considered biotic. Yes, Anita. Hydrogen bonds are between water itself and covalent. No. So you actually have those flipped. So covalent bonds are inside of water, and then the the covalent bonds are within water, but then there's hydrogen bonds between water molecules. So covalent you can think of as like inside a molecule. It's what makes a molecule, like makes it a molecule. Whereas hydrogen bonds are what exist between water molecules. What are the whole? Okay, so I think I just told you about the proteins, right? I mean, the idea they're made in the ribosomes, rough ER, Golgi, and then out. Now, of course, not every protein is going to do that. Some proteins just go to other parts of the cell and get used. Would a robot count as a living thing? Probably not, because again, you want to, well, not probably not, it wouldn't. Because if you think about the characteristics of life, it can't do all of those things. It can do some of them, right? But really, the important thing is that a robot doesn't have genetic material. A robot, it really can't really, you know, um, can't really evolve, not unless someone else does that for it. Um, okay. Let's see. Yes, and again, remember that for all fermentation, it's happening in the absence of oxygen, right? So the idea is ideally you use oxygen, get a whole lot of ATP out. But if you can't, then you're going to do fermentation instead. Let's see. Other things people are asking. To make sure that you can, I think one of the things you want to really make sure you think about is not just like memorizing terms or reading your notes, because yes, you could go through all these topics and be like, okay, I read all my notes. You really want to try quizzing yourself and applying the things, because we're not just going to say like, oh, just tell us the definition of everything, right? We might give you a scenario, and then when you have those scenarios, you have to apply what you're learning about these things. Are there any species that are, no, I don't think there's, like, every organism has a really important role in the ecosystem. So to say that an organism is the opposite of a keystone species is basically saying it doesn't have any importance in the ecosystem, which is not true, right? Everything has some importance, although some organisms might play larger roles in an ecosystem than others. But no, I would say there's not a species that's the complete opposite of a keystone species. But again, it could have a lesser impact on the ecosystem. That's possible. Let's see, what else? Let's see. I think earlier I was talking about blood type. Feel free to ask more questions, but yes, you could see multiples on the final, right? I mean, that's technically a multiple choice question. Okay, food web and food chain. Really, a food chain is like part of a food web, right? A food web is just more complicated because now you're talking about not just like a linear thing where like this organism eats this organism, eats this organism, eats this one, but rather more often than not, it's a food web, which is to say one organism might be eaten by like two or three different organisms, and then each of them is eaten by like two or three other organisms. It is possible to connect multiple food webs together. Yes, for sure. Right. You could have organisms. I mean, I think more often than not, it's probably just more like one big food web because they're connected. But I suppose it is possible to create, connect multiple food webs together if they have common aspects, right? I mean, then anything can be connected together if they have those common organisms. But like I said, I feel like then you almost just call it a bigger food web. But yes, they can connect them. Blood type. Let's talk about that for a second. I did notice people had some confusion there. So remember that the idea is that on the surface of your red blood cell, you have certain proteins. The A or the B antigen for A, B, and O blood typing, and then the RH antigen as well. Antigen, remember, just means protein, right? And then in the liquid portion of your blood, your plasma, you have antibodies to fight against everything that's not you. So if you have A type blood, then in your plasma, you're going to have anti B. And if you are RH negative, then you're going to have anti RH because you don't have RH, right? But then you wouldn't make the antibodies against something that's you. So we want to kind of remember that. What happens to the cells of a leaf if you crush it? They die. I mean, essentially, if you crush the cells of a leaf, you would probably burst the cell. And if you burst the cell, then it will not be functional anymore. What telomerase does is, remember, it adds on the ends of the DNA of the chromosomes. So the idea is every time you copy your DNA, it actually gets a tiny bit shorter because of the process. So telomerase, especially in cells that are going to become like gametes, you want to put on the full telomeres because otherwise they would just be wearing away their DNA. So telomerase adds back the telomeres to keep the cells um, essentially from self-destructing. Well, everything, yes. I mean, more or less, everything's there somehow. I mean, depending on the topic, some might be covered in greater, in more places than one. But yes, everything's on there, I believe. Can you compare the Rh antigen of bold type? I'm not sure what you mean, Arshia. You said, can you compare? Um, Basically, Rh is just another antigen. It's just part of a different system. It's part of, uh, plus or minus. Um, so if you're gonna, you could actually do one Punnett square. You have multiple traits, but we didn't talk about how to do that. So for our purposes, I would say you can certainly try, but I would actually probably more, um, I would be more inclined to say do it separately unless you happen to already know how to do a Punnett square, because the Punnett square now gets much more complicated. It's no longer a 2 by 2; it's potentially a 4 by 4, which we didn't talk about in class. Yeah, if you put nail polish on the stomata of leaf, you basically block it. And if you block it, there's no gas exchange. So CO2 can't get in and out, O2 can't get in and out, and that's going to impact photosynthesis. Um, do plants go through fermentation? I think it depends on the situation. I think, remember, plants probably don't, honestly, because plants have their own built-in source of oxygen, right? They can do photosynthesis. And so when they do photosynthesis, they are naturally going to generate oxygen from um, from from photolysis. So they're not going to have that same issue of fermentation because they have their own, they like make their own oxygen all the time. We can't do that, right? The only way we get our oxygen is by breathing in from our lungs. So yes, and so yeah. So yes, putting nail polish on a stomata of leaf would basically block gas exchange. Um, the properties of water are the ideas of like what are the things about water that make it really good in living things. So things like cohesion, which is water sticks to itself, adhesion, it sticks to other polar molecules. Those are sort of the major properties of water that we talked about, right? That it can dissolve things, it's a universal solvent, etc. Are the telomeres of of cancerous cells longer? They're actually not longer because cancer cells kind of exist in this weird, like, right on the edge kind of thing, right? Because they're dividing a lot. And remember, the more you divide, the more you whittle down your telomeres. But what they also do is they have telomerase active, so they're adding on a little bit. So it's almost like when I say they're living on an edge, right? I mean they're on the verge of apoptosis. They're on the verge of self-destruction. So they've worn away enough of their telomeres that it's short. It's not that they're longer typically, but they add enough onto those telomeres to keep it from self-destructing. So it would be like someone, it would be like something going to the very edge and then backing up a little bit and then going to the edge and backing up. Like they're always safe enough to not get destroyed, but not such that they're not going to have extra long telomeres. Is meiosis one basically mitosis? No. Well, it's not mitosis because what happens is very different. But yes, I mean, homologous, the major difference between mitosis and meiosis 1 is that you're splitting homologous chromosomes instead of sister chromatids. And then for meiosis 2, it actually is pretty much mitosis, right? You're doing exactly the same thing in meiosis 2 that you did in mitosis, just with half the DNA. There's no difference between synthesizing and creating lipids. It's the same thing. Hi, David. David, your live stream is tomorrow, though. Not that you're not welcome to listen, though. But yeah. Okay. Other questions? Let's see. Make sure you can tell the difference between haploid and diploid. I know that's something we covered kind of last minute, but And so don't get overly caught up in the details, but do make sure you know the difference. Um, there's a lot of vocabulary. I would say um, if you're not using your like flashcards or something else like that, you want to make sure there's a way for you to tell the difference between, especially like the vocabulary that's confusing, things like homologous chromosomes versus sister chromatids or mitosis versus meiosis. Um, why? Okay, so why are phospholipids smaller than amylase? Um, so basically, phospholipids. Well, amylase is a protein, first of all, and proteins are generally much larger. The smallest protein on record is like 54 amino acids, and again, that's the smallest one, right? So phospholipids are typically just one phosphate group and then a bunch of carbons, which could be quite a few carbons, but comparatively, it's probably not going to be 54 long. So that's why they're smaller. They're just physically made up of fewer atoms. Is it possible for a cell to have a fourth of um, well, it's possible, right? In the sense that I suppose you could have all these errors, but I don't think it would be viable. Like the cell wouldn't survive. It wouldn't have enough um, stuff it wouldn't have enough instructions. That would be like saying I want to have you know, I want to have a cookbook and be able to make dinner for people, but it's not going to have any stuff in there. Like the cookbook only has like 3 recipes. Okay, Rh factor, remember, is just another protein on the surface of your red blood cells. So really, when we talk about your red blood cells, we have 3 proteins in play. We have our A, our B, and then Rh, right? And the Rh is what makes it positive or negative. So whereas A, B, and O has to do with, there's Santa, Mr. Chow. Whereas A, B, and O has to do with codominance, right? You have A and B are codominant, but they're dominant to O. Then Rh is just going to be plus or minus, and that means that you're going to you either have it or you don't. And positive is dominant to negative. Okay. So, like I said, I think the tricky part is unless you know how to do those Punnett squares where you're doing the 4x4, you want to consider like A, B, and O blood type in one Punnett square, and then consider the Rh factor in a separate Punnett square, because really they're separate things. Okay, any other questions? Looks like we have a couple more minutes. What do I mean by lipids being packaged? Do you mean like what the Golgi apparatus does? Lipids essentially, proteins can be packaged. Essentially, they can be like modified, things can be added to them. Kind of like if you're going to get something ready to mail, you're going to, if everything you're going to put on there, then it just means you're sort of getting ready to mail, right? I wouldn't just put like something I wanted to mail in my mailbox without putting it in a box or putting a stamp on the box or things like that. Well, everything that we covered, yes, everything on the salmon sheet is what we've covered. So I was just trying to give you a better idea of what to study. Biomagnification is when you basically, as you go up the food chain, you have higher and higher concentrations of a toxin. And so it means that the higher you go up, because it's more concentrated, it's going to have a larger impact on the organisms there. And typically that has to do with the fact that, the organisms at the bottom eat just a little bit of the thing. So they have a little bit of toxin, but now everything eats more and more of what's below it. And so it keeps collecting.
Daddy, Butter Beans is on and I want to watch.
Okay, no, we already watched a lot of Butter Beans.
Butter Beans is on what I wanted to watch.
The difference. Okay, go get your snack. Go ask Daddy for your snack. So yes. So diploids, you have 2 sets of chromosomes, yes, and haploid, you have 1 set of chromosomes. What do you specifically need to know about pedigrees? I want you to be able to read a pedigree. I want you to be able to look at a pedigree and understand what's going on. So you should be able to figure out from a pedigree, for example, like what the genotypes of certain organisms, certain individuals would be, right? You should be able to say, possibly, depending on the pedigree, like, you should be able to take a pedigree and do a Punnett square and say, okay, well, if I see that these two parents have these genotypes, then I can figure out what possible genotypes the children could have. Autosomal means not sex linked. So it means anything that's on one of those 44 chromosomes that is not X or Y, that is autosomal. And so when it's autosomal, it just means your Punnett square looks like our typical Punnett square with just like big A, little a, little a, little a, right? Whereas a sex linked trait means your Punnett square now has to include the X and the Y's. Other details? Other questions? Then what are you eating? You say hi?
Hi. Hi.
Okay, go play, please.
Hi.
Okay, you show them your cookie? Okay, go play. Go play, please. Any other questions?
I like you, Mama.
Thank you, sweetie. Okay, I need to go there. Okay. You want to show them your ducky? You're going to show them your ducky and then you go.
What is it?
Mm-hmm. Okay. All right, go. Go color. All right. Any other questions? Go, Sammy. Come on, go. Thank you. Anabolic and catabolic. Okay. So for my students, that's not something you need to know. Anabolic and catabolic, it has to do with like reactions, like whether you're breaking up something or you're building it up. That's really what it means. So they're usually used to refer to like reactions. Okay, no, we're not watching anymore. Thank you. Let's see.
My orange juice.
So typically, anabolic involves like making things, whereas catabolic usually I think means like breaking it down. I think that's the difference. Desalination means removing the salt from something. And so technically it could be a way for us to get water, but it's such an expensive process that it's not really a way that we try to get our water at the moment. When given the phenotype of the child and the mother, yeah, the father can have multiple things, right? Because what you have to consider when you're trying to solve those problems is first you have to consider, what do I know about the child and the mother? If you're given a phenotype, let's just say that the child is like A negative, right? Well, A, first of all, and you have to take those two separately. A could mean AA or AO, right? But if they're negative, they have to be minus. On the other hand, if a child's like A positive, they could be like AA plus plus, or they could be AA plus minus, right? So I would start by saying, what are all the possibilities? But then it's like a puzzle, because now you say, well, if you now consider like, okay, now I know that the mother, let's say, is like A positive, but the child is A negative. It's not possible now for the mother to be plus plus, because when you do that, then the mother has to give one of her alleles to the child, so she has to give a negative one since her child is a minus, right? So yeah, you want to just sort of think about that, and then it probably would help you to like draw out the Punnett square and see, is it possible to get this as one of your products? If it is possible, then that could be the parent. If it's not possible, you rule it out. If given the phenotype of kid and father, yeah, in this case, it basically is saying, unless you're dealing with a trait that's on X or Y, it doesn't matter. Like, the idea is that if you have one parent's information, you could have more than one possibility for the other ones. Rishi's asking if there are 46 chromosomes, then why? So only 22 are numbered because the last pair are the sex chromosomes. So you don't really, I guess they don't really number it then, in the sense that like just called the sex chromosomes. How are the proteins made from a dominant different from proteins? Well, it really depends on the allele, first of all, right? But in the end, the idea is, and we'll learn about this more next semester, The idea is that the instructions for those proteins are slightly different. And because of that, there's a couple possibilities. It could mean that the instructions are slightly different, so you make two slightly different proteins. It could mean that in one case, you, it means in another case, it could mean like because the instructions are different, that like I'll just make this, it's one possibility, right? That the dominant allele produces a certain protein, but then recessive, because the instructions are essentially there's a mistake in them. What do you want? Okay, can you ask Daddy to help you guys now? Then could be now that like dominant makes a protein but recessive doesn't, right? You make like one makes a functional protein and one makes a non-functional protein. So there's not really a way to say like all dominant alleles it's this and all recessive alleles it's this. It's just the idea that they're different forms of a gene, right? So think of a gene as like a recipe for a cake, and there could be lots of different ways to make that cake. So depending on which cake you made, you're gonna have different results. Does A minus O minus mean A? Yes. So if it was AO, that means you're going to have. Okay, sorry about that. So yes, if you're AO minus minus, and your phenotype of your blood would be A negative. If someone has hemophilia, it means that you actually can't clot your blood very well. So it means, for example, that if you like normally when you get a cut, your body has a way of basically plugging that cut up really quickly, so you don't lose too much blood. But if someone has hemophilia, they can't do that, so they're much more at risk of bleeding to death, basically. Anemia is different, although they could be related. Anemia usually refers to how much. It has to usually do with like how many red blood cells you have, and because of that, your ability to carry oxygen in your blood is lower. So they both have to do with blood, but they're different blood disorders. Like one has to do with how well you can like heal after a wound, and then the other one has to do with your ability to carry oxygen.
My lips are red or no.
Okay, other things.
What color are they?
Would the amount of white blood cells increase the impact of AIDS on your body? Okay, well, basically, without getting too much into this, because we've not really talked about it, your white blood cells are part of your immune system, right? And so AIDS is a disease that attacks your immune system. So usually it's caused by the virus HIV, and the idea is that it attacks your immune system. And so when someone has AIDS, it means their immune system is actually so depleted that they have very few white blood cells left. So yes, in that sense, it definitely does. It's not that it increases the impact; it's that's what AIDS is. AIDS is essentially like your immune system is like gone, almost gone. Okay. What happens in interphase and mitosis? So interphase is really, there's two ways of thinking about interphase. Interphase, first of all, is just how your cells hang out and do things most of the time, right? They're not dividing, they're just doing their normal cell thing. But sometimes in interphase, one of the things they do in interphase is they do get ready to divide. So they're copying their DNA, they're copying the organelles, their cells getting bigger. They're doing all the things they would need to do to be able to split the cell in two and still have two complete cells. Whereas in mitosis, that's the physical act of splitting, right? So you could think of interphase as like the prep beforehand. Like if I know I'm going to have to take like one big box of stuff and split it in half so that I have two equal boxes now, interphase would be the process of like doubling all the stuff in my box, like making sure I have like two pens and two pencils and two pieces of paper, whatever it is, so that when I split it, I'm going to have one for each new cell. So interphase is really like prep for division as well as all the stuff in your body, whereas like mitosis is the actual act of splitting it in half. Are your white blood cells and antibodies similar? They're not similar because thank you, Sammy. So white blood cells are cells of your immune system. Some of your white blood cells. Okay, thank you, Sammy. Go. Thank you. Go play. So your white blood cells, there's actually a lot of different kinds of white blood cells. And so some white blood cells called B cells will create antibodies. And so it's not that so antibodies are proteins, basically, whereas white blood cells are cells, right? So there are some white blood cells whose job it is to produce a lot of antibody proteins. What happens if your cells are incapable of mitosis? Well, there's a lot of things, right? First of all, not every cell in your body is going to go through mitosis. It's not that they're incapable of it, but they don't always go through mitosis. But if your cells can't go through mitosis, then you'd have a lot of different issues, right? You wouldn't be able to grow.
Sauce.
You wouldn't be able
Cranberry sauce.
No, thank you.
Cranberry sauce.
Okay, Sam, you need to get down, please. This is not okay. Get down, please. Thank you. No, then no random talking. Thank you.
Sammy, come here.
So if your cells don't go through mitosis or cannot, right? It would mean you couldn't like fix your wounds. You couldn't. Oh, thank you, baby. You couldn't grow. You know, you just all the things where you might need more cells. You wouldn't be able to make more cells for all the reasons you might need to be able to make more. That you might want to make more cells. Okay, let's see what else. What's wrong, baby? No, it's okay. Any other questions then? Yes. Sorry, no, it's okay. You are correct. The difference between diploid and haploid is diploid has 2 sets of chromosomes, haploid has 1 set of chromosomes, right? Because the idea is that haploids are going to be the gametes that are going to become half of the one organism's contribution to the future offspring. So triploid is a thing. Yes, there are some things, there are some cells, some organisms who have triploid DNA. In fact, some have octoploid, some have like, septoid. I mean, there's something like that. The idea is yes, like so. Haploid means 1 set, diploid means 2, triploid means 3, right? So yes, their triploid is a thing. You don't see it in humans, at least not usually, right? But Samantha, please get down. Yes, this is not okay. Go sit down, please.
I'm gonna get something from my pocket.
Okay, that's fine. Let's see. Any other questions then? Okay. So haploid or diploid is just the idea of Lucy. Sex cells are haploid, right? Sex chromosomes is X and Y.
What does she do?
So in that sense, a haploid cell will divide all of the DNA, and one of the things that will be divided is the sex chromosomes. But sex chromosomes are X or Y. Okay, any remaining questions? Looks like I'm gonna have to go soon, probably in the next 3 minutes or so, because my little ones have to get ready for bed. So if you guys have more questions, so can someone be made of 2 eggs? No, I don't. I mean, well, not without like manipulation in a lab. So an individual, there is a way, like, when we talk about cloning next semester, like there is aspects of it where the cloning involves 2 eggs, but not 2 whole eggs, right? But could they potentially take, the sort of cellular stuff from 1 egg and put the DNA from another egg into it? Yes, they have done that. But from in nature, normally no, 2 eggs do not generate a new diploid organism. Any other questions? Let's see. Any other questions then? If you do think of other questions, like I said, you can email me. You can come see me during brunch. So
what's brunch?
Brunch is when they have a break. Say their snack time.
What's brunch? Mama.
No one's eating brunch right now.
When they get to do their playtime.
Well, everything that we will be covering. Yes, I mean, again, it's the bullet points are a guide, but you should check your notes. But yeah, everything's there. There's not going to be there shouldn't be something that you don't see there that's on the final. But again, it has to be application, right? So I mean, those are topics. It's not like you have to make sure you can take those topics and connect it to other stuff.
I love food.
Thank you. No, thank you, Sammy.
Because it says say something, so I had to say something.
Oh, okay. You guys are very welcome. Have a good weekend. Let me know if you have other questions.
I love Ducky.
Okay, thank you. Okay, Sammy, why don't you say bye-bye? Baby,
Hi,
say bye-bye.
bye. I love Ducky.
Okay, can you guys say good luck on your finals?
Good luck on your broke.
No, good luck on your finals. Baby,
No,
say bye-bye.
Bye-bye.
Okay.
Brow.
Okay, we better get these guys to sleep. So have a good night, everyone. I'll see you guys later. And again, just come find me if you have questions, okay? Or email me. All right. So long.
Bye-bye. I love Ducky, misters.
Okay, thank you.